Welcome to the first module of Introduction to Genetics. In this part of the course, we lay the foundations for your understanding of how genetic information is stored, transmitted, and expressed. We will begin with the historical roots of genetics, the groundbreaking work of Gregor Mendel, the structure and function of chromosomes and genes, and then move into the molecular processes that allow genetic information to produce the proteins that make life possible. Think of this module as building the framework upon which the rest of your genetics knowledge will stand. Genetics is not just an academic discipline, it's the very language of biology. The ability to interpret that language is essential for any biologist. 2. Historical perspective and Mendelian inheritance. Let's travel back to the mid-19th century, to the garden of an Austrian monastery, where a monk named Gregor Mendel was cultivating pea plants. Mendel was not the first to wonder about how traits are passed from one generation to another, but he was the first to design experiments that produced consistent, measurable, and interpretable results. Mendel's experiments. Mendel worked with pea plants because they had easily observable traits, such as flower color, seed shape, and pod color, and because they could be bred selectively or allowed to self-pollinate. He performed controlled cross-pollinations and carefully recorded the traits of the offspring over many generations. From his observations, Mendel deduced that traits are determined by discrete units, what we now call genes. These units are inherited in predictable patterns. Some traits are dominant, masking the appearance of others, which are recessive. Mendel's laws. From these experiments, Mendel formulated his laws of inheritance. Law of segregation: each organism carries two versions, or alleles, for each trait, and these alleles segregate during gamete formation, so each gamete receives only one allele. Law of independent assortment: the alleles for different traits are inherited independently of each other, as long as they are on different chromosomes or far apart on the same chromosome. Significance. These principles became the foundation of classical genetics. However, as we'll see in Module 2, there are exceptions to these laws. Nature has many variations and complexities. 3. Chromosomes, the carriers of genetic material. While Mendel identified the patterns of inheritance, he did not know the physical nature of the effectors he described. It was only in the early 20th century that scientists connected genes to chromosomes. Structure of chromosomes. Chromosomes are long, thread-like structures made of DNA and proteins, mainly histones. In eukaryotic cells, they are located in the nucleus. Humans have 46 chromosomes, 23 pairs, with one set coming from each parent. This diploid arrangement means each gene is represented twice, once on each homologous chromosome. Chromosomes contain DNA, the genetic material. Histones, proteins that package and organize DNA into structural units called nucleosomes. Centromeres, the regions that link sister chromatids and are crucial during cell division. Telomeres, protective caps at the ends of chromosomes that maintain stability. Types of chromosomes. Chromosomes vary by shape and function. Autosomes, non-sex chromosomes, which carry most genetic information. Sex chromosomes determine the biological sex of an organism, X and Y in humans. 4. Genes, structure and function. A gene is a segment of DNA that contains the instructions for making a functional product, typically a protein. Location and arrangement. Genes are arranged linearly along the chromosome. The exact position of a gene is called its locus. Functions of genes. Store information for protein synthesis. Regulate when and how much of a protein is made. Act in response to signals from the cell or environment. Genes can be coding, producing proteins, or non-coding, producing RNA molecules or regulating other genes. 5. Genetic combination. One of the most fascinating aspects of genetics is how variation arises in sexually reproducing species. Genetic combination refers to the reshuffling of alleles during reproduction, which increases genetic diversity. Mechanisms of genetic combination. Independent assortment. During meiosis, chromosomes align randomly, so each gamete gets a unique combination. Crossing over, homologous chromosomes exchange segments, producing new allele combinations. Random fertilization, the specific sperm and egg that meet contribute to variation. This variation is the raw material for evolution. Without it, natural selection would have nothing to act upon. 6. Molecular basis of gene expression. Genes are not static, they are expressed when needed. Gene expression involves two major processes, transcription and translation. Transcription, DNA to RNA. In transcription, a section of DNA is unwound. RNA polymerase reads one strand of DNA and synthesizes a complementary messenger RNA, mRNA, molecule. The mRNA carries the genetic message out of the nucleus to the cytoplasm. Translation, RNA to protein. In translation, the mRNA sequence is read by ribosomes. Transfer RNA, tRNA, molecules bring the corresponding amino acids. Amino acids are linked together in the correct order to form a protein. Regulation of gene expression. Cells don't express all their genes all the time. Regulatory sequences and proteins control when a gene is turned on or off. This allows cells to respond to environmental changes and to specialize for different functions. 7. Protein synthesis in detail. Proteins are the workhorses of the cell, performing structural, enzymatic, and regulatory roles. Steps of protein synthesis. Initiation, the ribosome assembles on mRNA and locates the start codon. Elongation, amino acids are added one by one to the growing chain. Termination, a stop codon signals the end, and the completed protein is released. Post-translational modifications, proteins may be folded, cut, or chemically modified before becoming fully functional. 8. Linking the concepts. In this module, we have seen how Mendel's principles describe the inheritance of traits, how chromosomes carry genetic material, how genes encode proteins, and how genetic combinations increase diversity. These form the backbone of classical and molecular genetics. In Module 2, we will go beyond the simple Mendelian view to explore the exceptions to these rules, how mutations alter genetic information, and how genetics applies to human health, populations, and microbes.